Hey everyone, welcome to Tricky Topics for Chapter 19. We're going to be going over recombination and transformation as well as dideoxy or Sanger sequencing. This video is going to be going through recombination and transformation, so make sure you watch that other one if you need help with dideoxy sequencing. So starting off with recombination and transformation, there is a huge long process to this, and before we go through that, I want to talk about what the end goal is because I think it can make it a little bit easier to understand as we go through if we have that end goal in mind. So our very end goal is to have a recombinant plasmid in a bacteria. And there's three main steps to this. The first one is to make that recombinant plasmid. And this process is called recombination when you're combining two different types of DNA together. The next thing we want to do is we want to take that plasmid and we want to put it in a bacteria. And the process of placing DNA into a bacteria is called transformation. Unfortunately, neither of these steps is guaranteed to happen. There's a couple other things that could happen as well, and you don't really have a way to tell which one is the recombinant plasmid in a bacteria, which one is our end goal. And so we finally, for our very last step, are going to have a selection process, in which case we're going to do some stuff, which we'll go over later, and we'll be able to pick out the bacteria with the recombinant plasmid, because that is our end goal. That's what we want to have. So we're going to go through each of these steps. Starting with step one, making a recombinant plasmid, we're going to be talking about the required materials. After we go through the required materials, we're going to actually go through the stages of the process. But there's going to be a lot of terms there, and so I want to make sure we really understand what each of those mean. So there's going to be four things that we talk about: restriction enzymes, also called endonucleases, restriction enzyme nucleases, plasmid, foreign DNA, and ligase. So let's start with restriction enzymes. We'll talk about the plasmid, foreign DNA, and ligase, and then go through the steps of the process. So restriction enzymes, or restriction endonucleases, they cut at very specific palindromic sequences. So remember with enzymes that they're each going to have a specific function based on what their structure is. So the sequence that they can cut at is specific because that's what their function is to cut at this specific sequence. And so each one would therefore have a slightly different structure. And they're going to be cutting at palindromic sequences. So in words that you guys are all used to, a palindrome would be whenever a word is spelled the same forward and backwards, like the word race car. In DNA, it's a little bit different. We call it a palindromic sequence if the strands are going to read the same if you read them from 5 to 3. So I gave an example of one here. On the top strand, you have 5' AAGCTT, 3', and we know the bottom strand needs to be anti-parallel and complementary to that. So the 3 and the 5 are switched, and then TTCGAA. But if we were to read that bottom strand from 5 to 3, you'd get 5AAGCTT, which you'll notice matches the top strand. So they're the same if you read them from 5 to 3. And restriction enzymes are able to cut at these sequences. All right. Now we talked about the restriction enzymes. Let's talk about the plasmid. So a plasmid is a piece of circular DNA, and there's 3 important regions that you need to know for them. First, you have the origin of replication. This is going to allow the plasmid to actually replicate. And eventually we're going to have foreign DNA in it. So it's really important for it to be able to replicate. That way we can study it and see what's happening. The next part is unique restriction sites. And each of those would be a palindromic sequence where a restriction enzyme could cut at. So you could use a lot of different restriction enzymes to cut at those sequences. And finally, you have selectable markers. And there's 2 that are super important. The first one is antibiotic resistance. And usually you're going to see ampicillin resistance, and we notate that with AMP-R. And the next selectable marker that's really important is the LAC-Z gene. So remember that LAC-Z is going to produce beta-galactosidase, and that is capable of breaking down XGAL. XGAL is a substrate that we're going to talk about later. And it's a substrate for the enzyme, and whenever it's broken down, it'll turn the bacteria blue. And the really important thing about the LAC-Z gene is that it's located in the same spot as your unique restriction sites. So if we were to cut, we'd be cutting in the middle of a LAC-Z gene. All right, so before we move on, let's draw a plasmid really quick so we're all on the same page. So it would be double-stranded circular DNA molecule. We have a origin of replication. We would also have an ampicillin resistance location. And we have our LAC-C gene, which throughout that gene, we're going to have our unique restriction sites, which I'm going to do in purple. And that's where a restriction enzyme could come and it could cut any of the sequence, and notice how they are located in your LAC-C gene. So let's move on to our next material, which is foreign DNA. Foreign DNA is going to be some long strand of DNA that within it, there's this sequence that we're interested in. And if we want this sequence, we need to be able to cut it out. So we know that one of the ways that we can cut is using restriction enzymes, and restriction enzymes need palindromic sequences. Therefore, we would want a palindromic sequence in front of and behind the sequence that we want to cut out, because once we do that, this sequence is free. So just to draw that out really quick, let's say that we have our double-stranded DNA, and there are 2 restriction sites right there. So we'll use whatever restriction enzyme we need to cut. And then you would have your foreign DNA fragment, which we would then put into a plasmid. And the very last material, just to talk about it very briefly, is ligase. Ligase is important in sealing the gap, because whenever you take the 2 DNAs and put them together, there will be a small gap, and we need ligase to create a phosphodiester bond between them. And that'll be in the very last step of recombination, connecting the foreign DNA in the plasmid and closing that nick. So now that we've talked about the required materials, let's talk about the actual process. In part 1, you're going to use a restriction enzyme that will cut at the same sequence on the DNA and the plasmid. The reason that we do that is if we use the same restriction enzyme, it'll cut at the same sequence, and then they'll be complementary to each other. So if you cut the plasmid, that'll cause it to open up. And if you cut a slice out of your DNA, you have this section right here, and we could put that into the plasmid because they're complementary. And that would be ideal. But unfortunately, that's not how science works for us every time. So there's actually 2 things that could happen. The first thing, what we want to happen is our foreign DNA, which is complementary to our plasmid, is going to attach, and ligase is going to seal the gaps. And this is called a recombinant plasmid because it has 2 types of DNA in it. But there's another option, which we don't want to happen, in which case your plasmid that opened is also complementary to itself. And it could just close up again without taking the foreign DNA in. And if it just closes up again and you don't add anything to it, that's called a non-recombinant or an intact plasmid. All right. And what's really, really important about our recombinant plasmid, so imagine on my hands that my fingertips right here are the LAC-C gene. If I were to open it up and just close again, my LAC-C gene is fine. If I were to open it up and now imagine that there's something in the middle right here, my fingertips are separated, meaning that my LAC-C gene was disrupted and it does not work. So just keep that in mind. We're going to talk about it more in a second, but keep in mind, foreign DNA in the plasmid would be in the middle of the LAC-C gene sequence, meaning it's disrupted and it does not work. All right. So that's step 1, and step 1 was all about making a recombinant plasmid. Now we have it, and remember our end goal is recombinant plasmid in a bacteria. We have a recombinant plasmid, let's put it in a bacteria. So remember that transformation is that process. And at this stage, there's 3 things that could happen. So remember, we had 2 types of plasmids formed. We had non-recombinant and recombinant. So 2 options are that the bacteria takes up the non-recombinant or the bacteria takes the recombinant. And our last option is that the bacteria just refuses a plasmid. It does not want one. So you have 3 types of bacteria: no plasmid, non-recombinant plasmid, recombinant plasmid. And with these 3 types of bacteria, if you're just looking at them, you can't tell the difference. You can't tell which one has a plasmid And of the ones that have a plasmid, which one is even recombinant? So we have a selection process, and there's two selection parts to this. The first one, all we're doing is trying to find which of these bacteria have a plasmid. At this point, we don't care if it has a recombinant or non-recombinant. We just want to get rid of any bacteria that doesn't have a plasmid. So every single one that we're looking at definitely has a plasmid. And the way that we're going to do this is with ampicillin. So remember, on every single plasmid, you had an ampicillin resistance gene. That means if that ampicillin resistance gene is in a bacteria, the bacteria would be considered to be ampicillin resistant. So if we use ampicillin, we know that ampicillin is an antibiotic that kills bacteria unless that bacteria is resistant to ampicillin. And we know that bacteria will be resistant if it has a plasmid, because every single plasmid is ampicillin resistant. So at this point in part 1, we're going to kill any bacteria that does not have a plasmid. So whenever we go into part 2, now every single bacteria has a plasmid. And now we're going to look at, okay, now we know they all have a plasmid. Let's try and find the one with the recombinant plasmid. So what we're going to use in this one is something called XGAL. XGAL is a substrate that beta-galactosidase, which is made by your LAC-Z gene, can break down. And if it breaks it down, it's going to emit a blue color, so your bacteria would appear blue. So if your LAC-Z gene is functional, we're going to break down XGAL and we're going to turn blue. If it's not functional, XGAL will not be broken down and your cell is going to stay white. So which one of these do we want? Well, remember we talked about earlier that our foreign DNA disrupts the LAC-Z gene, meaning if you have foreign DNA in right here, your LAC-Z gene is not working. And if your LAC-Z gene is not working, we're not breaking down XGAL and our cell is white. On the other side of that, if we had a non-recombinant plasmid where your LAC-Z gene, it was open, nothing came inside, so it just went back together. If it is working, that means that there's nothing disrupting it, meaning there's no foreign DNA. So at the very, very end, we would select for the white bacteria. All right, that was a lot. So I'm going to go through each of the stages a little more briefly and just draw it out. That way you have a visual as well. So let's start with step 1, which was all about making a recombinant plasmid. And remember that there were 2 options here, so I'm going to draw out 2 plasmids. And I'm going to draw some foreign DNA as well for each of these. So in step 1, we're going to take some restriction enzymes that are going to cut up our foreign DNA and our plasmid. So if I take my restriction enzymes, they're going to cut up my plasmid first. Or not necessarily, it doesn't have to be first, but we're going to cut it first here. And they're also going to chop up my foreign DNA so that now I just have a short sequence of it, and that sequence will be complementary to the plasmid because we cut it at the same spot. So one option is, since they're complementary, that foreign DNA is going to attach to the plasmid. The other option is that my plasmid says, I don't want that, and it goes back together. So we just formed 2 types of plasmids. We have our non-recombinant plasmid right here, and we have our recombinant plasmid right here. Step 2 is putting it in a bacteria. Remember, that process is called transformation. So I'm going to have 3 bacteria to show our 3 options. my bacteria could take up this recombinant plasmid, or it could take up the non-recombinant plasmid, or it could take up nothing at all, no plasmids. So if you're just looking at these, we can tell which ones have plasmids and which ones don't, but in real life, you want to be able to tell. So remember that on your plasmids, we have a couple different locations. So I'm going to go back to the plasmids now. On both of them, you have, do blue, an ampicillin resistance gene. And I'm just going to color the blue in here, but not label it. So both of those have ampicillin resistance. The other important point on our plasmids is our LAC-Z gene. So remember, for the LAC-Z gene, it's where my fingertips are. So if I go back together, that means my LAC-Z gene is still together and it's working great. But if it's my fingertips and my plasmid opens up and there's something that's inserted right here, I have LAC-Z on this side and foreign DNA in the middle. So that's what I'm going to draw on my recombinant plasmid. Where yellow is LAC-Z. All right. So now the first step is just trying to figure out which of my bacteria have a plasmid. And to do that, we're going to use ampicillin because 2 of these bacteria are ampicillin resistant, and they're the ones that have the plasmids. So if I put ampicillin in, wrong color, that's going to get rid of any bacteria that is not resistant, which is the one without the plasmid. And for step 2, I'm going to put in xgal, which, if my beta-galactosidase is functional, meaning LAC-Z is functional, it'll break down xgal and turn blue. If not, it'll be white. So this one that is functional, my second one, is going to turn blue because it'll break down xgal. My other one that's not functional will be white because I disrupted it. It doesn't work. And that is the process of having, end goal, recombinant plasmid and a bacteria. First make the recombinant plasmid, then put it in a bacteria, and then select for the one that you want. And that is the end of the video over recombination and transformation.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.